All right, AP Psych, we are in Unit 4, Sensation and Perception. This is video number 2 on vision. And so to start with, here's our diagram of the eye. Let's talk about what vision is going to sense in our environment, and that is light. And so when we talk about vision, we can talk about the wavelength and the intensity of light waves or light rays that we see. And so wavelength is going to determine the hue that we see or sense. And so color would be determined by wavelength. And, to review from science class, the wavelength is really just the distance from one peak to the next. Intensity, that's the amount of energy in the light wave, is going to be known as its amplitude, right, or height, and that actually determines the brightness of things that we see, or brightness of color. And so light is going to be our primary source of information concerning vision. The eye is going to be the structure that allows us to sense light. And when we talk about the eye, we can talk about a few different parts. The cornea is going to be the curved, transparent, protective outer layer of the eye. And to review from what you learned in video number 1, the cornea is the accessory structure to the eye. It's going to help the eye hone in on light sources. And so its curved structure helps focus the light into the part of the eye that needs it. The pupil, the little black circle in your eye, if you're looking in the mirror, is actually a hole in your eye through which light enters. The iris, or the colored part of your eye, is actually the muscle that adjusts the opening of your pupil. And so in a darker room, we will see the muscle dilate the pupil, and in bright, sunny, out-of-doors situations, you'll actually have your muscle contract and make that pupil smaller. The lens is actually located behind the pupil. It focuses images onto the retina, which is the inner surface of the eye. And so a muscle is actually bending it to curve more to keep close objects in focus, and to flatten out more to keep objects at a distance in focus. And so obviously those of us that wear glasses with lenses in them, or any of us that wear contact lenses, do so because of a faulty lens in our eye. It doesn't adjust to keep objects in focus as it should. Now, that sensitive inner surface of the eye that I had just mentioned is called the retina. That's light sensitive, and an outdated analogy is that it was much like film in a camera prior to the digital age. And inside your retina, we can talk about a couple of different cells. Rods and cones are the photoreceptors that convert light into neural activity. And so to review from your first video, photoreceptors are the sensory receptors in our eye, and this is where transduction is going to occur. We are going to see light energy being converted into neural activity. And so rods and cones are in charge of coding different pieces of information. Rods are not very color sensitive, but they are very light sensitive. Cones are going to actually have different photopigments in them that help us pick up differences in color in our environment. And so these photopigments are light sensitive, and when they're exposed to light, they begin to break down, which actually causes a change in the cell membrane and leads to action potential. And so rods and cones are those sensory receptor cells that translate light into a neural impulse that can be sent to the brain through the nervous system. Your bipolar cells are actually a special type of neuron that has two extensions. That's why we call them bipolar cells. And they kind of exist as the connection between the photoreceptors and the ganglion cells. And so they can act to transmit signals back and forth between the two. And so they're special neurons because they actually work on graded potential versus action potential. So all of that information concerning threshold and all or none response and refractory period don't actually apply to these very special cells. A good analogy is that bipolar cells are a lot like those interneurons in our brain and spinal cord, that they kind of act as the switchboard between the sensory receptors and the nervous system. And so they kind of have a special job but defy some of the rules of what normal neurons do. Your ganglion cells are the actual neurons that transmit this activity to the brain. And so the axons of all these ganglion cells form a fiber tract that travels from the eye to the brain. And we call that fiber tract the optic nerve. There is again, communication from those bipolar cells as to what information gets sent via the optic nerve to the brain. The last thing to mention concerning the eye itself is a blind spot, which is actually the part on the retina where the optic nerve leaves to go to the eye. And it's at that point that there are none of these photoreceptors or rods or cones. Okay, and so there's a part in your retina that actually can't see things. We all have these. It's not a mistake or a abnormality, and our eyes are constantly adjusting so that you don't even recognize they're there. But we'll do some activities in class to kind of show you what we mean by a blind spot. Fun historical fact is, legend had it, Charles II of England used to line the condemned up in his blind spot so that he could imagine what they looked like beheaded. And so AP Euro meets AP Psych right there for about 2 seconds. Okay, so concerning processing the information we get from our eye, some review from Unit 3, it's leaving the retina via the optic nerve, and it's first going to the thalamus, our brain sensory switchboard, before it is routed to that visual cortex, which is located in the occipital lobe. What is interesting about this process is that there are actually neurons in that visual cortex that are known as feature detectors. And these feature detectors receive information from individual ganglion cells that are traveling, from the retina. And they respond to specific elements of a stimulus. So like the outline of something, the color of something, the texture of something, whether or not it's moving. And so these cells then pass, or these feature detectors pass that information along to the proper cortical areas in the brain, or the proper cortices. And so, we don't really notice that because of parallel processing. And so this is review from an earlier unit, but if you recall, the brain can actually conduct separate tasks or analysis at the same time. And so when we see a bird flying through the sky, we don't really take note of its color, of its texture, of its outline, of the motion of its wings as separate pieces of information, because the brain is processing it all simultaneously. Okay, the only time you really notice a problem in a feature detector or notice their existence is when there is a problem with one. And so any of you that read the textbook section on this will recall Myers telling you about a Mrs. M who, after a stroke, couldn't see movement. And so I have some examples of things like that for you in class as well. That phenomenon is actually called blind sight, in which people are perceptually blind to a certain component of vision. Okay, and so I guess technically society labels them as blind in that they can't see, but really there isn't an issue with their eye or the optic nerve. It has to do with these various feature detectors in the visual cortex. And so some examples of people with blind sight, right? They have some awareness of the area, but they can't perceive what it is they're seeing. So we will do some examples of that in class and watch some videos related to that. Concerning seeing things in color, one thing to keep in mind is that this is science, not art class. And so when we talk about color vision, we are talking about the additive color mixing process, not the subtractive color mixing process. Okay, so if you look at the little picture there at the bottom right, you can see subtractive. That's like art class. When I mix all those different colors of paint together, I get black or a really dark brown. Or the same, like if I'm in a photography class and I'm hand developing color film, right? We would talk about the yellow, cyan, and magenta being mixed together to give us those colors. But concerning how our eyes work, it's actually, we're talking about light, and that when we mix all colors together, we get white light. And so white being the combination of all colors. And so keep that in mind, that the yellow paint in art class is technically absorbing all other light waves but yellow. So, right, the classic question on the playground, like, what color is the grass? And when people said green, nope, that's wrong. It's technically everything but green. Yeah, you can see why I didn't have a lot of friends on the playground. Anyways, I heart science, so who cares? Two other things related to color vision. Young and Helmholtz are going to come up with what's known as the trichromatic theory, and then Herring is going to come up with opponent process theory. So these are two competing theories that attempt to explain why or how our eyes see things in color. And so Young and Helmholtz come up with the idea that these cones in the retina that are in charge of seeing color actually have three different photopigments in them that correspond to the additive light process. And so they have photopigments for red, photopigments for green, and photopigments for blue, and that when they are stimulated by looking at things in the environment, whatever combination you're seeing, like reflected off an object, a different combination of the RGB photopigments in the cones are going to break down and cause that action potential. So that's one explanation of why we see in color. Herrings said that the chemicals in the cones are actually either working with one another or against one another. And so that's why you get the opponent process theory. And so he said that they would work in like opposites, like red versus green, yellow versus blue, white versus black. And so that's slightly different. In all fairness, color vision is probably a blending of those two theories. So I'll show you some examples of after images, which, like staring at something and then looking at a white sheet of paper and getting that ghost-like image afterwards. That's good support for Herring's opponent process theory as being a valid theory of vision. But we also have good examples of Helmholtz trichromatic theory being right as well, in that we can see, that there are examples of people who are colorblind that are missing specific photopigments in the eye. And so some examples here for you. These two fuchsia or magenta, purple, whatever you want to call them, squares, are they the same or are they different? The majority of you are probably going to say that they are the same color. Okay, in all honesty, I went in and adjusted the RGB on those pictures slightly, and so the one on the right hand side is slightly different in that it has less red, less green, less blue in it. So whether or not you could tell that it was different would probably depend genetically on how receptive or how acute your photopigments are in your cones. There are some genetic studies that show that women tend to have more sensitive photopigments concerning color than men, and there's a lot of good evidence to show that that the majority of people who are colorblind are male. And so the example there at the bottom is an example of what those of you that are see in full color can actually see that the picture on the right would be an example of somebody with color blindness, right? You're not blind to all color. You just can't discriminate some colors as well as others as a result of not having as many photopigments in your cones. So there you go. Lots of fun examples in class related to vision and visual processing. So thanks for listening.